afternoon, everyone, and welcome to the 15th MERLIN project webinar for 2023 on peatland restoration strategies. We are very privileged to have Professor Henrik, Dominik Henrik Zak from the Aarhus University in Denmark, who will be speaking to us.
Can I ask the selection of the plots? Is it random or cool?
The format is the normal format where there will be 30 minutes of presentation, and that'll be followed by question and answer. I invite you to submit your questions on the chat. I will try to keep track of them and put them on one document to keep track of who asked what question and then ask you in order to present your questions. So without any further ado, Professor Zak, thank you so much for coming. The floor is yours.
Yeah. Okay. Thank you, Jan, for the introducing words. I share my screen now. And you should see now the presentation modus, right?
Yes.
Yeah. And I hope everyone can understood me good, because I'm sitting here in my lab in Berlin in a moment, and there's an auto sampler making few noises, but not too loud, I would say. I hope you had a good summertime. It was a rather turbulent summer, I would say, in some places too wet, in some places maybe too dry. But now everything slows down a bit, I would say, at least in Denmark. I can feel that the autumn is coming slowly. And actually the slowdown, this should be a little bit the motto today for my presentation. And what I mean with slowing down is that we might slow down somehow our expectations in relation to the outcome of wetland rewetting or peatland restoration. And maybe we should also a bit slow down with our activities regarding the restoration of peatlands. And this is related to the water table rise. I will go through all these things you can see here on the first slide. Actually, there are 5 methods I want to introduce today, which is the just rewetting. And then we have opportunity to make topsoil removal. We can also do plant harvesting in the course of wetland restoration or peatland restoration, and we can make slow rewetting. And sometimes we do need a filter for phosphorus. And I don't want to say that there is one method or measure which fits to all sites. So really, we have to differentiate. And for doing this, we need a lot of knowledge, of course. And I will hopefully give you today a little bit of knowledge about processes driving, so to say, the restoration outcome, or better, driving the processes which result in a good or in a worse restoration outcome. And indeed, it is like this, that some sites which are just rewetted are characterized by an enormous high methane emission, or sometimes they release a lot of DUC or dissolved organic carbon, and some they release a lot of phosphorus. So actually these measures you can see beside of this just rewetting are aiming for reducing the so-called side effects. And I have a my presentation today will be divided in 3 main parts. I will give you a little bit of background, so about processes and reasons and then I will show you what we have done the last 20 years. And then I will go actually to the last 5 years and present what we have done here in Denmark to improve our knowledge in context of peatland restoration. And actually, I want to start my presentation with the conclusions, but the conclusions you can see here, they are old. They are almost 20 years old, and it is about 5 years after I completed my PhD. I was stating that we do need a complete rewetting immediately. And there were good reasons actually to say it, because we wanted to stop the peat mineralization taking place under aerobic drain conditions. So the demand was to make them completely wet to allow new peat formation once. But the point is, as I said before, we have the side effects. And actually, the upper image here, you can see it is not really looking different after 20 years of rewetting. Although there are some plants now inhabiting these shallow lakes, I was always stating, I was even writing it in my publications, iron-rich fens are the good fens, because those fens have a natural phosphorus trap. So the iron ochre you can see on the second image, it's a perfect phosphorus trap actually. So even you might have an enormous phosphorus release in the rewetted peat soil, the surface water will be characterized by very low P concentrations. But later we found that it's not only phosphorus we should focus on, there are other aspects. And we could see actually that the iron poor fens, they rehabilitate faster in terms of biodiversity. So we have to be careful with a statement, only consider iron-rich fens. And then you can also find in the literature the statement, and I was writing it also by myself, please avoid sulfate-rich water, because sulfate will, of course, push the phosphorus release. It's known since 100 years. But what we have seen actually, sulfate-rich water could also accelerate the restoration success. So you will hear about these three conclusions or how we have to deal with these conclusions more about the next minutes. Before, I want to give you the frame of our research activity, and you know it very well also from your countries. We have it in Denmark, in Germany, in the Netherlands, all over Europe and other parts of the world. We have this high nutrient loss from agriculture. We do have this high greenhouse gas emissions, and you know this alarming report showing us how dramatic the loss of biodiversity was within the last decades. And then there is the idea, there is one fit-all solution to cope all the problems, the nutrient loss, the greenhouse gas fluxes, and the biodiversity. And this is the rewetting of peatlands. Under natural conditions, indeed, there are perfect nutrient filters. They sequester carbon, and they can not only be very good habitats for a number of rare species, but they can also be positive, so to say, with adjacent lakes or rivers. But as I said, we not just press the water button and then immediately we have all this function in place. Although we can say it's not too bad. So we know that after rewetting, a lot of carbon will be sequestered, up to 700 kilograms per hectare and year. But we buy this carbon sequestration with an enormous methane emission. So the budget is actually that the sites are not really cooling, but they are warming. We also know that rewetted peatlands, they are perfect nitrogen removal agents, so to say. They can store or remove up to 100 and even more kilograms nitrogen per hectare and year, depending on the load and also depending some other factors. But as I said before, they can also release a lot of phosphorus. They can release a lot of ammonium, and they can also release a lot of DUC. So there are these side effects. And if you would be interested in birds, you would say, yes, this is the perfect site, because this shallow lakes are indeed very, very interesting habitats for a number of birds which were either completely disappeared or almost disappeared from the landscape. I don't know the names now, but all the people dealing with birds, they know they can be fantastic places. But actually, it is not really what we are aiming for. We wanted to have the brown mosses, the low sages, and maybe the orchids and so on. But they might last, I don't know, it might last 50, 100 years before we have them back. So this improving habitats is true for some aspects, but not what they were standing for before of drainage. And I want to say, of course, we not only have the wetland or the peatland rewetting in mind, there are also in Denmark other measures we're taking to improve the situation. So there is, of course, this constructed wetlands. We also alter the land use. And then, and this will be the topic today, we have this peatland rewetting or restoration. And the aim is in the moment that 100,000 hectares should be restored in the next years. And yes, just coming back to our expectations, there was indeed the wish 20 years or 30 years ago, we make them wet and then we have them back, this nice places with orchids and sages and brown mosses. But on the right-hand side, you can see what we have usually is a kind of biodiversity desert, so often dominated just by one halophyte species, like here, it is Typha. But I have to say, after 25 years, at least, we could see that Phragmites is coming into the sites and outcompeting Typha. And we have indeed also some sites where after 30 years, sage is coming back. And I'm always tending to compare something with humans, so to say, and some people do it also for lakes. I heard it before, that we have a sick, so to say, ecosystem. And what we see here, this high release of methane, the high release of phosphorus, might be translated to a kind of fever. And now you might argue, if you follow this picture, yes, just allow the sites to have the fever to become self-healed over time. But I think actually we not really can accept that this goes on for the next 40, 50 years, because as I said, there are the side effects which also deteriorating adjacent ecosystems. So I think we have to do something in some places at least. And I also want to highlight that we have a very good knowledge base already since really several decades. We know how the process is going into the soil regarding phosphorus cycling, regarding carbon cycling, regarding nitrogen cycling. And I don't want to detail it now. I will pick out a few of the segments here for later, showing later some results or drivers of the phosphorus cycling and carbon cycling. But I can tell you very short, we have, or we started now with having nutrient balances. We have a lot of research done on nitrogen and carbon cycling, even in re-wetted peatlands. We know a lot about phosphorus and iron cycling, and we go more and more into the water flows. But I think this will be the point which is most crucial. I will show you later why. And we just started now also with the biodiversity, which is actually really bad, I have to say. We had to start it earlier, but now we included also this important aspect in our investigations. And for all which are not so familiar, maybe with peatlands, you can see here the image of a really undecomposed peat sample. So if you drop into the soil, you can just lift it up with your hands. And what you can see is a really well-preserved organic matter. And there are some people, they are able also to say which plants were growing there regarding the structure and some elements in this peat. And there are some features I want to say to you, which are actually really, so to say, exciting for me always. The one is the high water content. So if this peat is water saturated, then the water content can be up to 99%. And you know what? Milk has a water content, has 95% or 97%, or in Denmark, you can also have some higher water contents. But the interesting point is indeed, despite of this high water content, you can work on the peatlands, but of course you have to be careful. And the other one, the other characteristic is the very high organic matter content. So the dry mass can be built up to 99% of organic matter. And this is the case for this peat sample. So these are really extreme features, so to say, of in peat soils, which also explain their important role in the carbon budget for all terrestrial systems. And what happens if the peat becomes mineralized? You can see it here on the lower image. I have to throw. You see that the peat is changing the structure. So it becomes very amorphous and you are not able anymore to recognize which plants were growing there. And this strange or this destroying of structure is of course accompanied also with a carbon loss. But still the carbon content is rather high. So for this peat sample, it was still 50% of dry mass. So it's not completely mineralized. It looks more like biochar or how you call it. It's just very humified material. And actually, it's not necessary that you make your fingers dirty. You should just see in the river valleys, and then you can often see this little molehills. And I don't know if someone is interested in gardening, because often the question rises, can we do something with this degraded peat? And I can tell you already now, I was testing it as flowering substrate for growing tomatoes. But this is something we learn later, why we should remove this degraded peat. Here, actually, I started my PhD. This is 25 years ago. And this is one site which is now a shallow lake since 25 years. And what we have done is we find out what is the peat situation in the whole 10 meters in this case. I was not digging down to 10 meters, but we were using a stake later on. But we could see in this site and also in many other sites that it's only the upper few decimeters actually which were affected by this drainage, which is a very interesting result actually, because the water table sometimes drop down to 1 or 2 meters under drain conditions, but still also in these cases which were heavily drained, the degraded, very humidified peat layer was in the upper 30 centimeters. And we still do this very simple method for the peat decomposition, and you will learn later why it is such nice and effective method. And here on the right-hand side, you can see the Sanico 25 years later. So there is still a shallow lake visible, but more and more the typha and the phragmites is going in and growing over. So there is a change, but the peat concentrations are still on a high level. The methane emissions goes a little bit down now, but DOC and also ammonium is also on a very high level, even after 25 years of rewetting. And I will show you very few results. We did a lab experiment in the beginning of rewetting to find out which soil layers contribute to the DOC release and to the phosphorus release. So we were taking a soil sample from the highly decomposed peat, from the lower moderately decomposed peat, and then from the lower slightly decomposed peat. And we were making them just wet over a time of 1 year. And then we could see for phosphorus, this was very interesting, that only the highly decomposed peat actually was releasing phosphorus. And you might now say, yes, this was only this site, Sanico, but I can tell you, we repeated these experiments in many, many other sites, and this was always confirmed. That's only the highly decomposed peat releasing this high phosphorus. And you can see it's a little bit different for DOC. Here we could also see that there's a little bit of DOC, or there is actually a large release of DOC also in the moderately decomposed peat. And you might now wonder what was taking or happening in situ. So in the site itself, I was not including it here, but I can tell you for the phosphorus, we were putting in dialysis samplers, recording the phosphorus levels in the soil water, and it was really exactly in between these two lines here, the black one and the green one. So the lab experiment, at least for the first year, was nicely mirroring the real P release condition in the wetland itself or in the peatland. I want to give you a little bit more theory. So you can see here a site which is nice orchids and also other plants, sages and so on, which is indicating that we have good conditions in this site. And this was actually true also under low drainage condition, means the water table was only dropped down by 20, 30 centimeters and then used as extensive meadows. But what we have done from the 60s in the last century, we were putting in this enormous drain ditches, and they were very effective transporting the water out. And of course, putting a drain ditch in a system like this can drop down the water table. And now I will disturb you with fluctuating water tables. And this is, of course, also fluctuating water tables are also disturbing the peat itself, because it brings oxygen in, and then the microbial activity will be accelerated. And then we have a transformation of this non-mobile phosphorus and carbon into mobile phosphorus and carbon forms. And always, if the site becomes wet, and this happens also under drain conditions in the winter time, in the wet period, then we have enormous leaching of nutrients and organic matter. And of course, if we have a complete rewetting, then all these redox-sensitive compounds, they will be dissolved, and this is explaining the high P release. I show you later a bit more on this. So here's just on the right-hand side, you can see the mobile P form. So we were quantifying the amount of phosphorus which can be mobilized from the degraded upper layer and from the less decomposed peat, and you see the big difference. And the question now is, how long does the phosphorus last? How long do we have to expect this high P mobilization? And what we have done is we have applied a chemical extraction method to quantify the so-called redox-sensitive phosphorus pool. And we have done this here for the MERLIN site, which is Corning. So more than 100 peat samples were taken from this site, and then they used the chemical substance to quantify the redox-sensitive phosphorus. And the result was per hectare, we have about 100 kilograms or mobile phosphorus in this site. And there were also a model developed to make a prediction on the phosphorus release based on this redox-sensitive P pool, or the iron P ratio in this pool. And with this P release rate, we could estimate that it would last about 6 years before this pool is completely exhausted. And I have to say, this is a very simple estimate, because it ignores, of course, that there's new phosphorus maybe coming in from the groundwater, and also other processes are ignored here. So it is just a very rough estimate. And I want to come back to this iron barrier. You can see here the phosphate, and in some cases, indeed, we found that the phosphorus concentration in the soil water can be up to 20 milligrams per liter. So it is comparable actually with wastewater. And actually, this was financing our research projects, because the agencies in Germany, they're noticing this very high pollution of water. And then they were, of course, afraid that there's a high P export to downstream systems. But we have to consider that not all phosphorus will be really exported to the adjacent system. There is this iron 2, which is oxidizing at the soil water interface, which is, so to say, a redox interface. And then most of the phosphate will be just trapped by this iron precipitation. And this process actually is also used in wastewater treatment plants. If they have still 50, 60, 70 micrograms per liter in the water, which is not good enough, then they add this iron 2 to come down to 10 or even lower micrograms per liter. And the good thing is we don't have to wait until rewetting to find out what will be the iron phosphorus ratio. We found that we can just make an investigation of the iron P ratio in the soil, and then we can predict the iron P ratio in the soil water after rewetting. So we have a very, very simple tool in place to make a rather good prediction if there is a P risk area, or if there is a lower risk of phosphorus export. And I also want to add here that this redox barrier is not only working for phosphorus, also a lot of DOC, which is released into the soil, will be trapped by this iron precipitation. So it's not so much as for phosphorus, so it was only an average 30%. But if we compare the surface water, we often see that the DOC concentrations are not so high in surface water, because there are other processes apart from this iron precipitation which drop down the DOC in the surface water, which goes then into adjacent rivers or streams. And the question which I was answering in the wrong way, so to say, before, is the sulfate pollution. So if there is a lot of sulfate maybe coming from groundwater or also from surface water by flooding events, then of course we know there is this sulfate reduction taking place in water saturated soils. And you know this hydrogen sulfide, which is the smelly substance from rotten eggs, this is very, very reducing substance. And if we have a lot of redox-sensitive PE in the soil, then this iron will be reduced and a lot of phosphate will be released. And at the other hand, also this process of vivianite, I don't know if you heard about this, which is an iron 2 phosphorus mineral, this will be also disturbed. And the result of the sulfate pollution is, yes, the phosphorus in the soil drops down faster and the phosphorus export to adjacent system might be faster, but the advantage is, of course, that the system becomes purified also faster. So if we have adjacent a stream or river, which is already very eutrophied, then we might accept that there is this higher sulfate-induced P mobilization in a rewetted peatland. But of course, it depends very much on which site you're talking about. And then in Denmark especially, but I know also in Germany and in the Netherlands, we have very nitrate polluted groundwater. And if we want to use now the term good or bad again, so you might argue from this scheme that nitrate is actually very good in terms of phosphorus retention, because nitrate is actually oxidizing iron 2 in anoxic conditions. And due to this oxidation, we have the formation of redox-sensitive iron 3, which is also in the soil a very good phosphorus trap. So what we have seen is if the water is nitrate polluted, then a lot of phosphate will be already trapped within the soil surface. So the peak concentrations are not 20 milligrams, but maybe 2 milligrams or lower. And then at the other hand, of course, also the P flux at the soil-water interface will be lowered. And we could actually also see that this nitrate, of course, it is increasing the mineralization, or let's say the respiration processes in the soil, but nitrate and this redox buffer function was also causing a lower DUC flux at the soil-water interface. And now it is a bit complicated, but you might heard from this very famous enzymatic ledge, which was published 2001, I think, from Chris Freeman and colleagues. And they said what you can see is all this carbon turnover processes. I don't want to detail, but they say there is this tannic or this phenolic substances we all know from red wine drinking. You know, this strange taste on the tongue. What is happening there? There are enzymes precipitated. And they said if we have a lot of tannins in our system, and this is typical actually for a pristine site, then the whole carbon will be just fixed in the soil because the hydrolysis of organic matter is, so to say, inhibited by the presence of these phenolic substances. And this study was conducted in bog systems, so in rainwater peatlands, and we were just curious to see if it is also true in calcareous or in mineral trophic peatlands. And we were investigating some sites which we were thinking are natural. And there are two included, Gützkow and Triebsch. We couldn't find the condensed tannins, which are known for inhibiting enzymes. And this is because, yes, they were also slightly drained in past. And but at the other hand, we could see that the degraded peat from rewetted sites, they had no condensed tannins at all. So maybe also this aspect indeed of missing condensed tannins also after 20 years of rewetting might be one reason that we still have this high respiration or release processes in the peat soils. And I come now to that what I actually wanted mainly tell you. This is our 5 management strategies, and I have translated now the pictures into symbols. So I will talk a little bit now about this inundation, about topsoil removal, about harvest, about slow rewetting, and also about the filter material. And the first question always is, can we do this restoration measures? And I know we can do. So there are examples from the practice where they have removed this degraded peat soil. I know few examples where they're also regulating the water table, but of course it is not always easy. And then I also want to highlight one more thing. We have a new boss term, so to say. You might say it in this critical way, but actually I like this nature-based solution concept very much. And there are maybe two big advantages at least. And the one is nature-based solution. It's just that you implement a method which is, so to say, copied from nature. And you not necessarily have to use this word restoration, because this restoration always implies that we find a natural system afterwards. No, the nature-based solution is just stating we applying a nature-like method. And the other thing is that with this new term, we have also a standard, so to say, which not only consider the site itself, but also all the things around. So there are social and economical aspects and other things included to make an assessment if we really have a good approach in place. And in the scope of this MERLIN project, I was doing this analysis for the Quarning site. And I could say, without going into details, that we are not too bad, but we have also some things to do further. And one point is, for example, this adaptive management thing. And we have also a nice development now in our group in Aarhus. We do have now several so-called living labs in place, which we densely instrumented to analyze, for example, greenhouse gas fluxes, to analyze water table changes, to analyze water quality changes, and even to analyze the fluctuation of a soil surface. And you can see they are spread over whole Denmark. It's only 10 sites, and I have to say not all 10 sites are densely instrumented. We actually have 4 sites which are a bit more instrumented to really go in detail with our investigations. And I also want to highlight one more thing. Before, actually the last 20 years, I was doing mostly biogeochemistry, which is interesting enough as it is climate research or biodiversity research or hydrology research. So every aspect can live for themselves. But the really nice thing is to combine it. And this is actually what we try to do in a moment, put everything together to have a better understanding of the processes, and above all for the release of phosphorus, methane, and other substances in such re-wetted peatlands. And what we also do now is, before, at least for me, it was true, it was only the molecular and the laboratory level. But as I told you, with the living labs, we have now the nice advantage to go also there. And we also more and more include now the remote sensing method to have a really nice overview, so to say. But of course, we know it that the uncertainty, the larger the scale is, becomes also larger. But there might be some improvement also with the methods in future. So this 100% uncertainty is maybe too pessimistic, and it will be definitely improve over time. And for the topsoil removal, just one result from this one site. I did some investigations where you can see here, if you are familiar with vegetation, after 6 years, this photo was taken, and you could even see this nice hollow structures. We could see the brown mosses and the low sages. And we also did some phosphorus analysis there in the soil water, and this was also on the natural level. So this topsoil removal was indeed a kind of reset in this system, I have to say. And the results you can see here on the right-hand side are from a bog system. So in the bog system, they also made a topsoil removal experiment, as you can see from this mineral trophic peatland, and they found that the methane emissions really dropped down by factor 100. So the topsoil removal can be a very efficient method, but I can say already it's not universal. And here are some other results. The numbers you can see here on top, this 174, 47, and 16 kilograms methane per hectare and growing season are actually related to a lab experiment. So what I have done is I was just going to different sites and harvested the dominating plants. Here it was Ceratophyllum, in the other case it was Typha, and in the next case it was Sages. And I was just incubating them under anaerobic conditions, and we could see that the plants alone could explain differences in methane emissions. And then we have a long-term study here in Sanico, and indeed the lab results were confirmed by this field study. The vegetation change we found in this site was accompanied with a clear drop down in methane release. And now the conclusion here is we can, with the help of or support of harvest and maybe also water table fluctuations or manipulations, we can indeed manipulate or influence at least the plant succession. So I want to say that we can manage indeed the methane emissions from such sites. And the number is 15, not only today for the webinar, but also true for an academic conclusion. You might heard about the water table must be -10 or -15 centimeters below soil surface. And then actually we have the best global warming potential effect after rewetting. And if you look into the next figure, you actually confirm this relation. So indeed, -15 centimeters below, it's also accompanied by low methane emissions. But if you go higher, it not necessarily means that methane emission must be high. So it can be extreme, yes, up to 2 tons per hectare and year. But in another side with the same water table, the methane emissions can be 1,000 times lower. So this overriding role of water table for methane emission really should take with caution. It's true for lower water tables, lower than 15 centimeters, but at water tables higher than 15 centimeters below soil surface, it is really scattering a lot. So there are definitely other factors we have to consider. And then there's another aspect, and I show you now one of our living labs where we simulated the water tables after rewetting. And you can see the more blue it is, the deeper the water table is. And this about 500 hectares. So there will be some sites which are inundated up to 1 meter, but there will be also some sites just in the neighborhood where the water table is still 50 centimeters below soil surface. So we have to be a little bit careful with our wish or dream of a constant water table. But still, we have the opportunities to make at least some manipulation, which is also useful. And in many cases, not in many cases actually, but in some cases, despite we have an enormous phosphorus removal at the soil-water interface, there is high P concentrations in the water outlet. So it can be higher than 100 or 200 micrograms per liter. And in this case, it might be useful, especially if the recipient downstream is a P-sensitive system. It might be useful to introduce a phosphorus filter. And we have started now with a phosphorus sorption experiment. And this was laker. I don't know if you know this material. It is used for hydroponic plants, for example. It's a clay material. And this had a P binding capacity of 1.5 milligrams P per gram dry mass. And then we knew that the P concentrations in the ditch could be up to, or on average, maybe 200 micrograms per liter. And we know that the water flow was 1 liter per second. And we were hoping that in this 1 second, 0.15 milligram will be retained, which we still have to prove, by the way. But if this is the case, then after 10 seconds already, that sounds not so much, but it's only 10 seconds, 1 gram would be saturated. And after 80 days, 700 kilograms would be saturated. It just want to illustrate, this sack was something like 300 euros. So it is very, very costly method, of course, to retain phosphorus from a ditch. And we still don't know actually how it really works in situ. And I want to give you a little outlook because we need still methods as simple proxies. The water table can be a proxy and the iron can be a proxy. And that's why we developed a device, I call it iron strips or iron filters. We developed, which hopefully we can use later as a proxy for making predictions of phosphorus and carbon fluxes and maybe also for biodiversity. But this is still what we are working on, but it looks good, at least from the side of reproduction. And regarding the first results we have found in situ, it seems to be suitable that we might use such an iron filter in the field to make some predictions regarding fluxes. And what we also apply now is this geophysics methods. I don't know if you heard about this TGM, and you can see here an ATV, it's tracking such a nice device over the field. So if you have 400 hectares, for example, after a couple of hours, you can make nice images. And these images are just standing for the resistivity, which actually means how good the water is flowing through the soil body. And actually, we were using those information for putting our piezometers in where we expected that water is flowing. And the next step will be that we might use such information with other additional investigations on hydraulic conductivity, and so to have a pedo transfer function to have a tool later to predict water flows, and also not only the where it takes place, but also regarding the velocity. And all what I have said now, it's actually combined not only in the EU MERLIN project, but there are other projects in total. It is indeed 12 projects which are dealing now with all these questions. There are still a lot of uncertainty. So we do a lot of monitoring now, which is really, really important for testing the different measures. We have a number of pilot projects where we in detail test the harvesting effects, the topsoil removal effects, and the phosphorus filters. And our dream actually is at the end of the day to have a toolbox in place you can use for certain sites to make predictions on nutrient release, carbon release, and so on. And for Denmark, at least, we're also planning to make a nutrient mapping, or just saying here are risk areas and here are low risk areas. And we will do it on the basis of soil samples which were taken before. So there is a dense network of samples taken before we might use later to use all the other information and making this kind of mapping. And I'm now to the end. Did I take too long? Yes, what's 40 minutes? Sorry, Jan. But I'm almost done. I want to say slow rewetting can be a good option, but we can see it is also a bit critical maybe in some places. I can tell you topsoil removal can in some places a perfect measure, but not always. And please consider iron not only be good, it can be also bad, so to say. And by the way, it can be also have no effect. And sulfate can be indeed a restoration driver. And what we still need is we need really the validation of few things by an ongoing monitoring. And all this, of course, is only possible if you have a good team. And so there was at least one wish becoming true. And we have a nice wetland group in place now in Aarhus, where we can combine different expertise regarding greenhouse gas fluxes, biodiversity, and so on. And here you on the right-hand side, you can see the names of our group. So thanks, I'm done.
So thank you very much, Dominik. Of course, the only one key reason why we try to watch the time is if there's not enough time afterward for questions, but you may be in that special category of such a clear lecture that no questions emerge. We have two questions, however.
Yeah.
And that is from Hassan. The first question is, what type of land use did you consider for your study? Example, if farmland or peat extraction site. And so does the type of land use before restoration affect the results of your analysis?
And Jan, can you repeat what type of? I didn't understand this.
Sorry, what type of land use did you consider for your study? The examples is farmland or peat extraction site.
Okay, yes, it is indeed, it is both. Sorry, I have.
Would you stop sharing your screen,
Yeah.
so I might be able to see if people raise their hand?
Yes, I stopped sharing, right?
Thank you.
So we consider both or several land use types. In some cases, it is indeed also peat extraction sites. We have one site which was used for peat extraction. And we have also sites which were used as arable land before. So there was maize or other crops cultivated. And actually the most sites we investigating were used as grassland before. So either as extensive or as intensive grassland. And indeed it is it makes sense to differentiate here. So you cannot compare, so to say, a peat extraction site with an extensive or with an intensive grassland site, or with a site which was used for crop production, or where ploughing was applied. And so you have to be careful what or how it was started. It is really a good point to consider.
Mm-hmm. The second question is from Linnea Yule Nielsen. How have you implemented biodiversity into your work as of now?
Yeah, it's a good question. It's not very, very smart for now, but what we started now is we recorded the plant species growing before of rewetting, and which is the plant succession. So this is what we have done regarding plants. And what we also do a little bit now is we include microbiology. So we also see how the microbial communities are changing, and we compare it also to pristine sites. This is also very interesting, actually. I thought it is boring, I have to admit, in the beginning, but now I much understand few of the results or of the fluxes we measure much better in context of microbial changes. And actually what we are completely missing, which is really bad actually, is also to include insects, for example, because this is a very exciting topic, I would say. So butterflies or maybe also others, which would be worthy, I think, to see how they are affected by our restoration measures. So yeah, we are just in the very beginning, and we have also not so much expertise in the moment in our group, but we do quite good collaboration now with people from Greifswald and also other institutions to fill this gap, and hopefully also we will maybe do a little bit more in our group in future. So it is open, I would say, and there's much to do in this context.
Thank you. We have another question coming in. I want to encourage people to please put your questions in the chat, or you can just simply turn your video on and raise your hand. I'll recognize you and you can ask the question yourself. The next question is from Peter Duran. How slow is slow rewetting? How does this compare to simply letting ditches naturally fill in over time?
Yes, it is a very good point. And you know, I have to say that this is not my idea, and it's not a new idea. I know a few nature conservation groups, they have applied it already very successfully. What they have done is they were not maybe necessarily switching the pump station or were closing the ditches from one day to the other. They just allowed, so to say, a slow water table increase by doing by just stopping the maintenance of the ditches, for example. So I actually talk about a time frame of 10 to 20 years. And what they found is indeed, I mean, I have to say they were not doing detailed investigations regarding phosphorus or other things, but what they have seen is that the biodiversity was very well developing, so to say. So rather than having a shallow lake with typha or with phragmites or even with hydrophytes dominating, they were ending up after 10 to 20 years with rather good composition of at least plant species. But I don't know so many examples. So actually we are also here in the very beginning, and I cannot say 5 years this water table, 10 years this water table. But at least in Germany, we have the opportunity indeed to make such a water table management, because they have this wire system, this complex ditch network. And here you can indeed manipulate water tables. And in Germany, there is a kind of categorization of water levels. I don't know now, not each category, but there's very deep drainage and slightly drained and moderately drained and so on, which stands for a certain average water table. And the idea is that we maybe use this ditch wire pump system to really try to rise the water table, maybe in the first 5 years by, let's say, 50 centimeters on average, then the next 5 years maybe by another 50 centimeters on average, and then maybe after 10 years we just stop the maintenance of the water table management. And we not only do the water table management, we also think we should combine this with plant harvest to remove the nutrient surplus from the sites. So the combination of slow water table rising and nutrient removal by continuous of harvesting might be a very good method to improve the restoration outcome. But we don't have really good data to validate those things in the moment.
Thank you. We have a question here. Someone, I'm not quite sure how to pronounce their name. I can spell it. It's AU749046. And the question is, how many years on average for carbon neutrality?
Oh, yeah. I heard terrible numbers last time from someone, and it was indeed several hundred years. But we have to be careful because this has been really very degraded sites, and it has been also mineral trophic sites. So I don't know how it is in bogs. I could imagine that in bogs it goes much faster, I would say. But in mineral trophic, highly degraded, so intensively used sites, we might really wait not only 10, 20, 50 years. It looks like it could be up to 100 years and longer before they become neutral again. So it's a really long time span, so to say.
Okay. Well, again, it seems that you said things so clearly that we only got 4 questions. Unless someone has another question, I'd like to take a moment before everyone may come up with a question to announce that there will be the next webinar on the 2nd of October. And this will be the results from MERLIN's work package number 4 on working with economic sectors. I will, unfortunately, will be traveling in Asia at that time, so someone else will moderate. And that'll all be, you'll see announcements about this from the MERLIN project. I would thank you very much for a very clear, well-illustrated lecture, Dominik. And I see also Jörg has put in that you can revisit this lecture with if you go to the website, it has been recorded and you can see the entire lecture again. So thank you again. And for everyone, we'll see you in October.
Yeah, many thanks also from my side. Bye-bye.
Thank you. Thanks a lot.
Bye.
Have a nice day.
Bye.
Yep.
Thanks, you
Bye.
too. Bye bye.